good to go. Do you have the full screen or do you just see the slides? I'm not quite sure what you're seeing at the moment. Or
You need to play the slides because we have the full screen.
okay, great.
You're perfect.
All right. Okay, well, thank you all very much for being online today. We've got about an hour here to go over what is a pretty broad subject dealing with microbial systems that both produce and consume or biodegrade tertiary butyl alcohol. Just so you know what you're actually looking at in this cover slide, this is actually a bottle of tertiary butyl alcohol, which is one of the strange things. It looks kind of crystalline, and if you've ever had this material arrive at your lab, you'll sometimes be a bit concerned about, oh, it's a solid. Well, that's what it looks like. It tends to freeze at a pretty high temperature. Okay, how do I move my slide? There we go. So what we're going to talk about today, I've broken this webinar up into sort of three sections. And we're going to have to paint this in fairly broad strokes because of a fairly limited amount of time. But first of all, what I'm going to talk to you about are what do we actually know about tertiary butyl alcohol production and consumption. So we'll talk a little bit about the features of TBA. Obviously, a lot of what we know about TBA is related to the biodegradation of MTBE and other fuel ether oxygenates such as ETBE. And then I'm going to sort of walk you through pretty quickly the distinguishing features between aerobic and anaerobic MTBE and TBA biodegradation. Next section, I'm just going to present in very summary form some new observations. Several of them have come from our lab, dealing with strange or unusual or perhaps unexpected sources of tertiary butyl alcohol. First of all, I'm going to tell you a little bit about some work we've done with MTBE co-metabolism by ammonia oxidizing and methane oxidizing bacteria, two organisms or groups of organisms that haven't been identified as MTBE degraders previously. We've also done a lot of work on the biodegradation of isobutane. There's another potential source of tertiary butyl alcohol. And then there are a few other perhaps anecdotal sources of TBA which are worth mentioning. We'll briefly talk about them at the end of that section. Then the last section, I'm not quite sure whether it's named properly, what is unresolved, but really what my intention was to describe what the strengths and the limitations of the various molecular and chemical methods that are available for detecting MTBE and TBA biodegradation in field samples. So this is not really a review of the techniques so much, but it's really trying to explain where you're on good solid ground and perhaps where you're on less solid ground using some of the molecular techniques, qPCR, etc., or some of the chemical techniques such as CSIA. Okay, with that in mind, let's move on. So this first slide just gives you an idea of what tertiary butyl alcohol is. It's one of the 4 butanol isomers, all of which is shown there. It is the simplest tertiary alcohol. Goes by a whole series of other names, 2-methylpropanol, terbutanol, etc. Its physical properties, though, it's very, very water miscible, infinitely soluble in water, has a very low volatility and limited absorption to organics. So very distinctly different from a lot of the gasoline components that it's often found and associated with. The major sources of TBA obviously are the biodegradation of ether oxygenates, so both the aerobic and anaerobic degradation of MTBE, methyl tertiary butyl ether, and ETBE give rise to TBA. And as such, it's TBA's accumulation is often seen as a prima facie indicator of the degradation of those ether compounds. Unlike MTBE and ETBE, tertiary amyl alcohol, tertiary amyl ether gives rise to tertiary amyl alcohol, TAA. So we're primarily looking at issues that impact MTBE and ETBE. I mentioned earlier that TBA is sometimes seen as a prima facie indicator of MTBE degradation. That's not strictly true. Obviously, there are some complications in there. One of them is that tertiary butyl alcohol is actually present in fuel-grade MTBE at a low, but can be quite significant level. And TBA was also used as an independent oxygenate, entirely separate from its as a co-product or a co-contaminant with MTBE in gasoline. So that was in the late 70s, early 1980s. And then the other major common source of tertiary butyl alcohol is as a co-product from propylene synthesis, propylene oxide synthesis. So the reaction at the bottom shows you we can take propylene and isobutane and generate propylene oxide as the main product, and TBA as the co-product. So these are the structures of the compounds that we're interested in. Here's methyl tertiary butyl ether. There's the tertiary butyl group. There is the methyl ether group. Here is its kissing cousin ETBE, same tertiary butyl group. And here we have an ethoxy rather than a methoxy group. And both of these will biodegrade into tertiary butyl alcohol. Over here we have the structure of tertiary amyl methyl ether and undergoing the similar sort of demethylation reaction to give you tertiary amyl alcohol. So in very, very broad strokes, these are the biodegradation trends that we see for MTBE and TBA out in the environment, or whether it be in field samples or microcosms taken from field samples. These are the various terminal electron acceptors that, we're interested in the environmental field. So we're going from carbon dioxide here all the way through to oxygen. These get progressively more and more oxidizing as we go from left to right. And these are the products that we generate as a result of the reduction of these compounds. This one over here is kind of important. This stands for acetic acid. So what we are looking at here is the production of acetate using carbon dioxide as electron acceptor. This is, as you'll see a little bit later on, it's a very important process as far as MTBE and TBA production are concerned. So if we look at the general trends, as you go progressively more and more oxidizing, so the degradation rate for most chemicals, and that would include MTBE and TBA as well, increases as you go from the reductive environment over on the left-hand side through to the oxidative environment over on the right-hand side. So you get more energy, you get more biomass. Unfortunately, because MTBE is associated very, very closely with gasoline, the opposite is true in terms about the frequency of conditions that you find in gasoline-impacted sites. So you're putting a readily biodegradable organic in the form of gasoline into a contaminated site. All of the available electron acceptors often get consumed, and we end up with a highly reductive environment. Okay, so if we then look through the assembled data that's out there, microcosm studies, field studies, there is data that suggests that MTBE will biodegrade under all of the conditions that we have listed here, whether they be highly anaerobic through to highly oxidative over here on the right-hand side. In contrast, the same is not true for tertiary butyl alcohol. Certainly on the more oxidative end, there's very, very clear evidence for oxidation of TBA under aerobic conditions. And in general, the evidence gets progressively not weaker, but less substantiated as we go towards the reductive end of the spectrum. So, for example, there are a couple of recent studies that have really sort of, in the last year or so, have confirmed, perhaps unequivocally at the microcosm level, that we can see biodegradation of TBA and mineralization of that compound through to CO2 under sulfate and ion reducing conditions. What is clear, though, is that out in the very most reductive environments, TBA appears to be a stable compound. So let's have a little look at why that might be. So I'm going to go through anaerobic MTBE biodegradation separately from anaerobic TBA degradation, and then I'm going to switch over to the aerobic processes. I think it's always worthwhile qualifying what the information from microcosm and field studies said. There's a lot of variability out there, but in general, these microcosm studies and field studies suggest that MTBE production of TBA is being seen under all of the purported electron accepting conditions that we just listed in that last figure. In the case of the lack of TBA degradation under highly anaerobic conditions, the argument is that the organisms that we are looking at that are responsible for MTBE degradation are acetogenic organisms. And this is work that's come out from Max Hagbund's lab, Rutgers. The idea essentially is that here we have MTBE. The organisms that are responsible for degrading the MTBE essentially are the masters of C1 chemistry. They will remove this methyl group, and it is used with CO2 to generate acetate. Hence, these organisms are acetogenic organisms. And once that methyl group has been removed from the MTBE, we're left with TBA as a spent byproduct. There is no further biodegradation of that compound by these organisms. There's been reports of MTBE's degradation under anaerobic conditions associated with methanogenesis. Almost certainly that process is due to the fact that there are a lot of methanogenic organisms that can then take acetate that's generated by acetogens and then cleave it to make methane and CO2. So that's the prevailing, perhaps the strongest model for anaerobic MTBE degradation. When we look at anaerobic TBA degradation, again, there's a lot of variability and inconsistency in some of the data, but it now appears, I think, fairly solidly that iron reducing, sulfate reducing, nitrate reducing systems have the capability to biodegrade TBA. Again, it's variable. I think the second point that I make here, that TBA biodegradation may be more sustainable with higher versus lower concentrations, is really partially common sense. It's kind of what we see with MTBE as well. If you have low ppb concentrations of these contaminants, it requires an energetic input from the organisms. They have to be able to get a return on their investment for synthesizing the enzymes that are necessary for them to biodegrade these type of compounds. And when you're in these low ppb scenarios, sort of 10 ppb or perhaps less, the amount of energy that can be obtained by these organisms is very low. And so while you may see biodegradation of TBA at a higher concentration, ppm levels, where it represents a really viable energy source, that's a less reliable issue at lower concentrations. And again, there's some work that's just been published from Kevin Finneran's lab and also from MI, dealing with isotopic forms of TBA, which show that we can get mineralization of TBA under this sort of interface between iron and sulfate reducing conditions. And organisms like Geobacter, perhaps unsurprisingly, have appeared as potential candidates as the primary TBA degraders. Okay, so we're now going to switch over to aerobic conditions. And I think before we sort of dwell on that, go into it in any detail, it's very important that you get an idea of what the distinction is between the various biodegradation mechanisms for MTBE and TBA that are out there. The first one is a strictly metabolic process. So in this case, we'll have organisms that will take MTBE or TBA, and then they will biodegrade them through a series of very highly defined intermediates and ultimately generate carbon dioxide as the terminal product. And so the carbon and energy obtained from that process is used to increase the number of cells and we get biomass. So this is the classical biodegradation model of increasing number of cells as the compound is biodegraded. The type of organisms that catalyze this process, the two best known ones are methylibium, petroliophilium PM1, and Aquincula tertiary carbonis L108. For convenience sake, I've just put the genus name and the strain number down here. But these are organisms that will grow readily on MTBE and TBA. The fairly limited distribution of these organisms, or perhaps limited distribution is the wrong way to say it, there are a limited number of examples of these organisms, which then leads you to suspect that they are of limited distribution. The second biodegradation mechanism that applies both for MTBE and TBA is co-metabolism, and this is frequently seen in alkane oxidizing bacteria. So an organism that can grow on an alkane will take that alkane, oxidize it to an alcohol, oxidize it to an aldehyde, an acid, and eventually through to carbon dioxide. And so it is actually growing on that compound. And then the degradation of MTBE and TBA is effectively superimposed on that underlying growth process. And the degradation process relies on enzymes that the organism is expressing primarily to oxidize these compounds. It just happens to be able to oxidize some of these compounds as well. So this is referred to as co-metabolism. The vast majority of organisms that biodegrade MTBE co-metabolically generate tertiary butyl alcohol as the product, and some organisms will take those metabolites and further oxidize them. Key species that you need to keep an eye on would be Pseudomonads, Pseudomonas menacina, Pseudomonas putida, etc., and various Mycobacterium strains. So, if we have a look at aerobic MTBE biodegradation, certainly what we know is that TBA is an obligate intermediate in the pathway of MTBE metabolism. So you give organisms like PM1 and L108, you give them MTBE, they will make TBA, but that TBA typically does not accumulate extracellularly because that's actually what the organism is growing on. And they will process that through to CO2, and TBA will not accumulate to any great significant concentration. Typically, those organisms use two different enzymes to oxidize MTBE and TBA, and you'll see the significance of that hopefully a little bit later on. When we switch over to the co-metabolic process, we find again that TBA is a consistent product in MTBE oxidation. This is particularly being well studied in organisms that grow on gasoline range alkanes, in the C3, C4 through to C8, 9 region. Although there are other organisms that can co-metabolically degrade MTBE to TBA. Because this process is superimposed on another underlying biochemical pathway, what we find is that TBA typically does accumulate. So it's a distinguishing feature between metabolism and co-metabolism there. Although some of these organisms, particularly the mycobacteria, can then further oxidize this TBA, and they often use the same enzyme system that they use for MTBE oxidation. So if you compare these two panels, what you will see really is the distinction between these two these two physiological processes is that in both cases TBA is generated. Metabolism, you don't typically see long-term accumulation of TBA, whereas in co-metabolic systems you do. Dealing with aerobic TBA biodegradation is a separate issue from MTBE. My take on the field is that TBA is generally reviewed as more readily biodegradable, well, as readily biodegradable under aerobic conditions. It's not particularly difficult to find and isolate aerobic TBA oxidizing bacteria. All of the model MTBE metabolizing strains like PM1 and L108 will metabolize TBA, but there are clearly examples of organisms that will grow on TBA but don't have the ability to grow on MTBE. And so that's worth bearing in mind. The nice thing about these rapidly growing TBA oxidizing bacteria is that we know a lot about the pathway of MTB oxidation or TBA oxidation. And so in some cases, we have genomes available for the organisms. In other cases, we have very, very specific gene sequences available for key reactions, which is very, very useful for the site characterization, identifying, quantifying organisms in field samples. And the last point that I draw your attention to is that there are some unusual and very distinguishing activities that are associated with TBA degradation under aerobic conditions. There are unusual enzymes and unusual products which can be potentially used to track and identify either the process or the organisms responsible for this in field samples. Okay, so having sort of given you that very, very broad overview of sort of the conventional wisdom, let me just introduce to you a few recent findings about some other organisms that can generate MTBE, that can generate TBA. Some of them do it from MTBE. Some of them do it from an alternative source of TBA, which is isobutane. And then lastly, I'll talk about some non-microbiologically related issues, some of these compounds down here, which you might encounter and might represent sources of TBA in some of the environments that some of you may work in. So let's deal with this first one. I've had a long-standing interest in ammonia oxidizing bacteria, otherwise known as nitrifiers. And these organisms are in many ways very, very similar to methane oxidizing bacteria. In both cases, these organisms are ubiquitously distributed. You can find them everywhere. We exploit them, for example, in wastewater treatment systems if we're looking at nitrifiers. Methanotrophs, obviously a very, very big deal for dealing with methane emissions. We find them in landfill covers. We can find them above gasoline impacted sites that are generating methane, etc. And these are the pathways and the enzymes, systems, intermediates that these organisms generate. So dealing with the ammonia oxidizers first, they convert ammonia through to hydroxylamine. This reaction is catalyzed by an enzyme called AMO, which stands for ammonium monooxygenase. And this is a very, very non-specific copper-containing enzyme. And then if we look at the methanotrophs, there are the underlying pathway of methane oxidation in methanotrophs is the same. We take methane, we oxidize it to methanol, we make formaldehyde, we make formate, and eventually convert it to CO2. The initial reaction in this process is catalyzed by an enzyme called methane monooxygenase, and it comes in two flavors. There is a particulate version and a soluble version. The particulate version is very, very similar structurally to AMO, and it's another copper-containing enzyme. SMMO, in contrast, is an iron-containing monooxygenase, and is less frequently or less widely distributed than the particulate form of this enzyme. So one of the things that we find is that both AMO and methanotrophs expressing soluble MMO can both oxidize MTBE. The surprising feature of this is that despite the fact that PMMO and AMMO are structurally very, very similar, we see no activity for PMMO for MTB oxidation. The rates of these processes are pretty low, but you know, they're readily detectable. One thing that suggests that they're unlikely to be particularly significant in the environment are the high KS values for these. So these are, I apologize for doing this in microbiological or biochemical terms, but these are substantial concentrations of MTBE. So these organisms are not doing this willingly, I guess would be the way to describe it. And again, like a lot of the other organisms out there that can generate TBA from MTBE, none of these enzymes or organisms expressing these enzymes can further oxidize TBA. Second subject I'll tell you about is microbial isobutane oxidation. So isobutane is a fairly common component of hydrocarbon sources, whether it be gasoline, natural gas, etc. And the underlying premise behind the work that we've been doing on this is, well, was originally prompted by the question, well, if we take out MTBE from gasoline, are there any alternative sources of TBA in that? And really, this isobutane represents an interesting chemical for that process. So, in a lot of hydrocarbon impacted environments, the organisms that biodegrade the gasoline components, they're doing it under aerobic conditions. They incorporate oxygen into the target compound, whether it be propane, butane, etc. And once that oxygen has been incorporated, the compound itself then becomes very readily biodegradable. These enzymes lack what is called regioselectivity, so they can actually hydroxylate or introduce oxygen into different positions on the molecule. And so here is our monooxygenase enzyme up here. It is obtaining oxygen from the atmosphere, and this is the substrate, isobutane 2-methylpropane. And sometimes you can introduce oxygen into a CH bond on one of the terminal methyl groups, and that will result in the production of isobutanol. And alternatively, you can introduce an oxygen between the carbon hydrogen here to produce, excuse me, TBA. And so this is what we have been studying for the last year or so. And so I'll give you a summary of the results that we have got from this type of process. So first of all, we're going to look at metabolism. So bear in mind again, what we are looking at here are organisms that are growing on this compound and using it as a carbon and energy source for growth. And we have compared the same organism, this is an organism I've worked with for a long time, JOB5, growing on isobutane either under carbon limited or oxygen limited conditions. This represents the isobutane concentration, so you can see over time the culture consumes it. You can see the concurrent decrease in the amount of oxygen. But the key thing to remember here or see is that we actually have quite a lot of oxygen left in this reaction at the end of the growth process. And during the growth of the culture, we see the production and then subsequent degradation of tertiary butyl alcohol. So this is under carbon limited conditions. If we now put more carbon in than oxygen, we see the same sort of process, but the end result is a little different. So here is the isobutane being consumed, here is the oxygen being consumed, but now the TBA that gets generated, we get a lot more of it, roughly 4 times the amount, and secondly, because the oxygen has now been consumed, that TBA is stable. And so when we look at this, we are looking at roughly a 1% efficiency in the conversion of isobutane through to tertiary butyl alcohol. And we have found that that number is fairly consistent for a lot of organisms that have the ability to grow on isobutane. Normally, I wouldn't report a 1% conversion efficiency as a particularly dramatic or exciting thing, but certainly when we started looking at isobutane co-metabolism, that number changed dramatically. So the data I just showed you there was for Microbial back here, JOB5. It grows on isobutane, and it's got a roughly a 1% TBA to efficiency of converting isobutane to TBA. And it does have the ability to further oxidize TBA. However, if you look at all of these organisms down here, all of these, with the exception of this Graphium fungal species here, all of these are organisms that can oxidize, co-metabolically oxidize isobutane, and rather than generating 1% of the total flux as TBA, these numbers range from 35 almost up to 100% for some of these enzymes. Add on top of that, none of those organisms have the ability to further oxidize TBA. So if these organisms are growing on some of these compounds, they're exposed to isobutane, they will fortuitously degrade it and generate TBA, and this is the rough proportions that uh, we're concerned about. So I bring that to your attention, two of these particular types of organisms to your attention. So number one, I've got down here Pseudomonas mendocina KR1. This is an organism that grows on gasoline range alkanes. And then secondly, Methylocyanus trichosporium OB3B, which is a classic methanotroph, which grows on methane. So I want to just give you a couple of examples of or schematics about where this might be significant. We'll start with the gasoline one, I think. So here is a potential model for the impacts of isobutane that are present in gasoline. It can be somewhere in the region of between 1 and 4 volume percent of certainly in winter fuels where you need a high volatility fuel. The possibility exists that we have a gasoline spill. This is from your underground storage tank. We have hydrocarbons, very volatile hydrocarbons, moving up from the LNAPL, being met at a point where oxygen is now, there's sufficient oxygen present. These organisms will be able to take isobutane and convert it to TBA, which is effectively taking a very poorly soluble volatile gaseous compound and turning it into an infinitely miscible water soluble compound in the soil horizon, and then that TBA will partition into water, and you could effectively end up with a contaminated groundwater source with gasoline that contains TBA but actually contains no MTBE. The second one was dealing with methanotrophs. And so while we've been interested in TBA production and its association with gasoline, our attention has progressively moved towards what we see, or the potential for TBA contamination in natural gas production environments. So this is natural gas components. So we've got methane. This is the main feature of natural gas. Typically looking at something between 70 and 90%. Then there are the longer chain alkanes, ethane, propane, and the butanes, both normal and isoforms. And collectively, these can represent somewhere in the region of 20 volume percent of a low methane containing natural gas. And then we have a series of other compounds, obviously CO2, oxygen, nitrogen, etc. So there are different flavors of natural gas, obviously. Sour natural gas contains a lot of hydrogen sulfide, hence its name. Conversely, sweet natural gas contains little hydrogen sulfide. Dry natural gas is mainly methane, but certainly given the current glut of methane that's on the market in the US, there is a lot of attention placed on sources of natural gas that are wet. In other words, contain high proportions of ethane, propane, and butanes. And so if you think about this from the perspective of a microorganism, you have a gas supply which is potentially being released into a virgin environment. It's going from an anaerobic system up to potentially an aerobic system. And there's plenty of methane around, which will be sufficient to grow methane oxidizing bacteria. The question then becomes, well, what can they do to some of these other compounds? And so this is some data that we have for organisms, methanotrophs, that grow, obviously growing on methane, and we then expose them to isobutane. And this is the time course of production. So this is the production of isobutanol, so 2-methyl-1-propanol. And this is the time course of production of TBA, 2-methyl-2-propanol. And this represents the fraction of the total products which were TBA. So we add this, and then ask what is the fraction of this. And it stays consistently at 65%. So these organisms are quite happy growing on methane. They give them a small amount of isobutane. They will convert it very, very readily, either into tertiary butyl alcohol or isobutanol, and they do this at a pretty rapid rate, and with a consistent stoichiometry. We do find that this reaction only occurs with SMMO. We have not seen any activity with PMMO. So in many ways, it reflects what we see with MTBE oxidation as well. And here we have a potential model for this. So here's a stray gas coming from perhaps a hydraulic fracturing system or a gas distribution system, for example. You have isobutane with methane present. That gas reaches a point where oxygen is available. We then convert that isobutane from a gaseous form into an infinitely soluble water form, and then we get groundwater contamination, TBA, with no evidence whatsoever of MTB oxidation around. And this could also potentially occur in the saturated zone. And I have certainly been working recently with folks in the Midwest states who are trying to explain how they are seeing tertiary butyl alcohol in some of these natural gas production systems without any evidence for MTBE or conventional sources of TBA. The last two things then in this section are some perhaps curious aspects or curious features of sources of TBA. One of them is tertiary butyl acetate, which I've abbreviated as TAC. There's certainly evidence from the patent literature that tertiary butyl acetate was at least considered and potentially deployed as a tetraethyl lead extender. So this is very different from EDB, for example. This is actually put in to extend the functioning life of tetraethyl lead, likely to have only been in super premium gasoline. The reason why we're concerned about it is that the half-lives for hydrolysis for tertiary butyl acetate, so it hydrolyzes the TBA and acetate, are particularly long. So this gives you the range for pH 7, 8, and 9. We're measuring these half-lives in years as opposed to days or minutes, etc. So a contamination that occurred from this compound in the 60s at a reasonable ambient pH could represent a long-term source of TBA. This is an ESTCP report describing the use of tertiary butyl acetate for cleaning applications, in this case for cleaning hands. Perhaps more significantly, this is a bit of advertising copy taken from Lyondell, who are a major producer of tertiary butyl alcohol. We now have tertiary butyl acetate as a VOC exempt solvent. And so if this solvent happens to make it into an environment where it will hydrolyze, either biologically or abiologically, you then have another potential source of tertiary butyl alcohol. Okay, so in the last section, the very last one then is tertiary butyl hydroperoxide. Tertiary butyl hydroperoxide over here is a very powerful oxidant. Gets used in the chemical industry as a radical initiator. I'm sure many of you are aware of this very controversial and many think highly flawed study by the EPA about the impacts of hydraulic fracturing on deep groundwater at the Pavilion site in Wyoming. Irrespective of which way you feel about that, one of the compounds that was detected at high concentrations in this environment was tertiary butyl alcohol. It was only detected in one of the two wells that were investigated. As part of this draft report, the EPA suggested that one potential source of TBA might be MTBE. The other was that tertiary butyl hydroperoxide is used as a gel breaker for hydraulic fracturing fluids. And this is an image that I borrowed from the Halliburton website that shows one of their hydraulic fracturing gels. And this gel material obviously is not particularly good for allowing free-flowing gas in the environment, so you need to break that up. And one of the ways that you can break it up is to include tertiary butyl hydroperoxide as a gel breaking agent. Okay, so there's perhaps some curious parts of the MTBE TBA story. Here are some of the caveats, the strengths and the weaknesses, and the various tools that can be used to detect these processes out in the environment. So I have generally broken this up into molecular methods and chemical methods. And so in this case, I'm really sort of focusing on what could we do with processes like qPCR, microarrays, stable isotope probing. And this section mainly concerned about compound-specific isotope analysis, but also this possibility that butene production could be a useful measure of MTB and TBA oxidation in the environment. So let's deal with MTB oxidizing enzymes first. Obviously, if you're going to do something in the molecular level, it's great to have a gene sequence so you can then do qPCR to quantify which organisms you are interested in. Well, here we have got a fairly comprehensive, not complete, but a fairly comprehensive list of the various organisms that are known to biodegrade MTBE, or at least major physiological groups of organisms. These are the various enzymes that they use to do this process. This indicates whether or not a gene probe or a gene sequence is available for that enzyme. This is the reaction that they catalyze, and this is whether it's the underlying process is metabolism or co-metabolism. So certainly there are genes available for organisms that grow on MTBE, and certainly I have seen studies where this has been the only thing that people have concentrated on. Is there evidence for alkane hydroxylase or cytochrome P450 as an indicator of MTBE degradation in the field? I'm personally not a great fan of 16S ribosomal RNA-based analyses because these enzymes and these genes can be traded around amongst organisms very readily. And a 16S ribosomal RNA sequence is only going to tell you whether the particular organism you're interested in is in there. It doesn't tell you really what it can do. So I am much more a fan of targeting the functional genes responsible for that activity. The downside to that philosophy, I think, well, there are a large number of MTB oxidizing enzymes out there. So which ones do you go for? Some of them will tell you whether or not you're seeing MTB degradation through a metabolic process. Some will be able to tell you whether co-metabolism is an important process, potentially. We can also distinguish several enzymes or several processes which, for example, Mycobacterium JOB5 actually converts MTBE to tertiary butyl formate, and the conversion of tertiary butyl formate to TBA is an abiotic or a primarily abiotic reaction. So depending on what question you're asking, there are genes available, but the take-home point, I guess, would be that there isn't a single definitive gene sequence, whether it be 16S or a functional gene that you can tap into that says, well, this tells me that I've got MTB biodegradation. When we deal with TBA oxidation, the same issue about 16S ribosomal RNA, I think, still holds, that this capability is fairly widely distributed amongst different organisms. And so there is limited value in saying, well, this group of organisms are present, therefore we've got evidence for TBA oxidation. Certainly under aerobic conditions, there are some very, very well characterized enzymes, which I think make excellent probes for qPCR or microarray based systems. And so without trying to overwhelm you with the biochemistry, this is the pathway of MTB oxidation or TBA oxidation, going from TBA through to 3-hydroxybutyrate. These are the types of enzymes that catalyze these reactions. So we have a monooxygenase, 2 dehydrogenases, and then a mutase. And then these are the enzymes, and they would then have corresponding genes that can be useful targets, which are really unequivocally related to TBA oxidation, or as unequivocally related to TBA oxidation as you can get. Certainly this enzyme system and this enzyme system are consistently associated with TBA metabolizing strains like L108 and PM1. This last reaction, this conversion of 2-hydroxy isobutyrate through to 3-hydroxybutyrate, is a very unusual reaction, and I would argue is the defining reaction of aerobic MTB TBA metabolism. So if you were trying to find a very precise gene probe for perhaps using qPCR, this would be probably the one that I would pick. The role of these enzymes and the genes associated with them, I would say, is less clearly resolved at this point. When we get to anaerobic MTB and TBA degradation, we're really at the opposite end of the spectrum. We have got microcosm field-based evidence for the degradation of TBA under these reducing conditions. Same is true both for MTB and TBA, but the thing that is effectively killing us in the molecular field is that there are no organisms available in pure culture. And so there are no 16S ribosomal RNA genes that can be used in qPCR-based assays, and there are no functional genes available that can be used in the same way as I just, for example, recommended or suggested that you could track that mutase for an aerobic process. So in the absence of those genes, in the absence of 16S ribosomal RNA or functional genes, the best molecular tool, the best one that we have currently, is to do stable isotope probing with C13 labeled substrates. And I'm just going to very briefly go through this. The whole idea, this is a fairly sort of startling image, I know, but the whole idea behind this is that the isotopic composition of an organism is going to reflect the isotopic composition of what it eats. And seeing as carbon is the predominant atom in biological systems, we focus on carbon. So the take-home point being here that under normal circumstances, if you feed an organism that contains a carbon-containing compound, and that carbon-containing compound has got natural an isotopic distribution, roughly 98.9% of that will be carbon-12, and roughly 1.1% of that will be carbon-13. And to all intents and purposes, that's what the isotopic composition of the organism will be. Same isotopic composition. However, if you artificially enhance the concentration of C13, that organism, when it grows, will reflect that isotopic composition. And so this is how we can do stable isotope probing to find out what organisms are there and which ones are operative. Very briefly, stable isotope probing concentrates on 2 different types of molecule. In bacteria, polysaccharides are what make up the cell wall of the bacterium. We're not particularly interested in those. Underlying that, there is a phospholipid layer, which makes up the membrane, which separates the cell from the internal part and the external part of the cell. Very interested in that. We'll talk a little bit more about that. In the nucleic acids, this is really what we're looking at here. This is the hard drive. This is the hard drive of the organism. Particularly interested in DNA. Less interested in RNA. So we just concentrate on phospholipids and DNA. Phospholipid fatty acids, you add C13 labeled compounds into a contaminated environment, and you're adding a C13 labeled contaminant of concern. If the organisms are present that can grow on that compound, the C13 label from that chemical probe will be incorporated into newly synthesized phospholipids. And then we can then extract those phospholipids, hydrolyze them, derivatize them, and then analyze them by mass spectrometry to determine whether or not we have got C13 incorporation that has gone from the contaminant that we used into the microorganisms that have utilized that compound. And so that is a very powerful technique, can potentially provide you unequivocal evidence for biodegradation, for example, and can some of the specific phospholipid fatty acids, these tails here, or what we actually analyze, can tell you which particular broad groups of microorganisms may be responsible for the biodegradation process. We can do something very similar with DNA. In this case, we're adding a C13 labeled compound into a sample, whether we do it in the field or a microcosm. The organisms that grow on that compound will synthesize C13 DNA. It will reflect the isotopic composition of what it was grown on. We can separate that DNA out by cesium chloride density gradients centrifugation. So this heavy DNA comes from the organisms that have metabolized the C13 compound, and this comes, the C12 DNA comes from organisms that haven't. And then we can molecular look at 16S ribosomal RNA content in the C13 DNA to identify organisms that have utilized that labeled compound. One way you can do this is to do it in a biotrap, and certainly there is information, I think, available on the MI site that will talk a little bit about how you can do this in the field, adding in a C13 labeled compound into these beads, which are then deployed in a well, and the microorganisms that we're interested in actually grow on these on the surface of the beads and then can be analyzed. Some of the caveats associated with that, obviously very water-soluble contaminants like TBA can actually leach out of these beads at perhaps less than ideal rates, and that can then lead to higher ambient concentrations than you might want or expect, or perhaps don't necessarily reflect the ambient conditions in the aquifer around the trap. So there are certainly a couple of publications out that describe recognize this impact and provide tools and explanations about how to deal with it. So if we look at the stable isotope sort of toolbox, we've talked a little bit here about phospholipid fatty acids, stable isotope probing, and DNA SIP. The other tool that's out there is compound-specific isotope analysis, CSIA, and the key distinguishing feature of this is that we're dealing with low natural isotopic composition of contaminants rather than systems where we're increasing and adding artificial concentrations of C13 labeled compounds. So in the last couple of slides, I'll just quickly go over how and why and whether or not CSIA is useful for TBA production, etc. So just in summary, CSIA is really a gas chromatography technique, a very sophisticated gas chromatography technique. If we're looking at the carbon isotopes, we can take a sample of groundwater, we can remove the contaminant of concern by purge and trap, put it onto a conventional GC column, and we can separate out our contaminant from other contaminants as a conventional chromatogram. What makes CSIA then unique is that it will take each individual peak here, put it into a combustion oven where the carbon is there is converted to CO2, and then the CO2 is then placed into an isotope ratio mass spectrometer, and we can look at the various carbon isotope masses in there and work out what the isotopic composition of the starting compound was. We can also do something very, very similar for hydrogen, except rather than combusting things to CO2, we're pyrolyzing things and converting everything to H2. And so the mass spectrometer here is looking at hydrogen isotopes. So why is this useful? Well, this gives you a little explanation of how or why CSI works. Biological systems fractionate carbon, which basically means that it discriminates against compounds that contain C13. Bonds that contain C13 are a little bit stronger than carbon-12. So while the biodegradation process occurs or a reaction occurs, it happens slightly faster with carbon-12 than it does with carbon-13. And so I have this image here, somebody trying to push a ball up a hill. This is carbon-12. When you switch it over to carbon-13, you can still do the reaction. It just takes a little bit more energy, a little bit more push to make it happen. And so over time, you see a fractionation event. And so here we see the total concentration of contaminant decreasing over time. However, the ratio of yellow C13 is increasing relative to the amount of blue. So even though the mass is going down, the ratio is changing and the sample is getting heavier. And so as things get heavier, it's an indicator that they have been biodegraded. When we look at CSIA, and MTBE, this is the sort of conventional wisdom at the moment. There are aerobic processes that show both a hydrogen and a carbon isotope effect. So the aerobic process cleaves a carbon-hydrogen bond, which is what we see here. We see a small fractionation for carbon, so as the compound biodegrades along this line, we're seeing a small effect from carbon, but we see a very large effect on hydrogen. Anaerobic processes cleave a carbon-oxygen bond, and so we see a large carbon fractionation, but a very small hydrogen fractionation. And then the abiotic acid hydrolysis is somewhere in between. Well, this is the conventional wisdom. This is probably perhaps what the world really looks like. This is some newer evidence that came from Monica Russell, dealing with the isotopic fractionation that occurs for a whole range of microorganisms. Again, this is for MTBE. There is the aerobic sort of window. This is the acid hydrolysis window. And certainly what the paper that Monica put out in 2012 demonstrated is that some of the aerobic processes for MTBE oxidation have isotopic signatures, 2D isotope signatures, which are almost identical to what you see for anaerobic conditions. So the conventional wisdom about CSIA for MTBE might need a little bit of reinterpretation. Not forgetting that this seminar really, or this webinar, is about TBA. I just need to give you a little bit of an update about where CSIA stands for TBA. In general, it is much, much less studied than MTBE oxidation and has really only been examined for carbon isotope ratios, not for hydrogen isotope ratios. And part of the reason for that is that the alcohol group on TBA has an exchangeable proton. And that exchangeable proton means that it can potentially impact the isotopic fractionation, the hydrogen isotope fractionation, and make the data very difficult to interpret. So that's probably one of the reasons why TBA has not been looked at. Another reason why TBA has not been extensively examined for CSIA is that the enrichments that do occur appear to be very small. So TBA consumption seems to result, under anaerobic conditions, seems to result in very little C13 enrichment. And these two graphs here are some data for a couple of organisms, or well, same organism, but a couple of different tertiary alcohols that we've examined using C13, CSIA. And again, these are the enrichment factors that we're getting for the degradation of these compounds. And they are very, very small and effectively well, not particularly useful for the field environment. The very last possibility then is if those systems aren't going to work, one idea that has been thrown out that hasn't been evaluated as far as I'm aware in the field is that butene production could represent a measure of TBA oxidation, certainly under aerobic conditions. And so you remember hopefully this pathway here. Well, it turns out that this enzyme also catalyzes an accidental or a seemingly physiologically irrelevant dehydration reaction, and the small amounts of isobutane get generated during the metabolism of TBA. And it has been suggested that the accumulation of isobutane in the subsurface might represent a measure of ongoing or historical TBA oxidation. Again, that has not been tested out, but is a possibility. Okay, so I know I've occupied a lot of your time. I think I'm pretty good, about 1 minute past. This is my summary here. Point number one, not all TBA is necessarily associated with biodegradation of MTBE and ETBE. We talked about tertiary butyl acetate, potential for tertiary butyl hydroperoxide, also the possibility that isobutane oxidation from gasoline or natural gas might contribute to TBA signatures in the field. It's not all about MTBE and ETBE, I guess would be the take-home point. Um, Certainly, our understanding at the molecular level and the microbial level, aerobic processes far outstrip anything that we know about anaerobic processes. And so consequently, we have a large number of gene targets that can be used for characterizing TBA oxidation and production. Perhaps the downside of that is that we have got almost too many targets. Um, and so really, I think if you are using molecular tools to try and interpret what is happening to TBA in the field, you have to be very precise about the question that you are asking, because we do have so many molecular tools that could be applied to answer that question. Certainly when we get away from aerobic processes and we get to anaerobic processes, I would argue that stabilized isotope probing is the current best available EMD for characterizing this process. The incorporation of C13 label into either lipids or DNA really provides unequivocal evidence that you have got biodegradation of the compound occurring, and potentially gives you target organisms that you could track if it was necessary for your site. And last but not least, CSIA is currently not useful for TBA degradation. And I would also add that results for MTB oxidation, in the light of some of the more recent findings, need to be treated with caution. And I think with that, appreciate you all hanging around. I know I've run over a minute or two. I'd be happy to answer any questions, and I think Dora is going to handle that for you.
Thank you very much, Mike. That was a very fascinating presentation. As always, I learned a lot from your presentation. So we do have some questions from the audience. From a practical perspective, do you see particular advantages to using pure oxygen over other air-based techniques like biosparging when we're trying to do MTBE and TBA bioremediation?
Okay. I can't say that I have a huge amount of practical experience in doing in addressing that in the field. Um, there was historically some suggestion that MTBE oxidation under aerobic conditions was a sort of super oxygen intensive process. I don't think that has borne out over time. So I obviously the cost issue has got to be in there. There is certainly some microbial data that suggests that some strains like L108 are capable of oxidizing MTBE at very, very low oxygen concentrations. And so you could perhaps extrapolate that to say that putting in pure oxygen might be overkill. Um, I personally, I think a lot of it would have to do with the cost of the system, and I don't think there's a magic, if you go to pure oxygen, it's going to work better, personally.
Okay.
Hope that
Next
helps.
question. I think that's good. So if TBA can persist in the environment for as much as 135 years and it was used as an additive in the 60s to 80s, is there a way to differentiate the additive from TBA due to biodegradation so that TBA can be used to potentially date a release? And if so, what method would you find to be most useful?
Okay, can you say that one again? It was tertiary butyl acetate, right? It's got a long half-life.
Okay, so maybe
Yeah.
that's where the confusion is on this, right? So do you think that there's a way to differentiate what's happening with TBA, what's from biodegradation versus what may be an additive?
Oh, okay. So the question is, can we tell the difference between TBA that you find in an environment that might have come from tertiary butyl acetate or might have come from MTBE, ETBE? Is that I
Yes.
suspect that's what the question is answering. Um, well, certainly from ongoing, well, okay, step number one would, I'd see if you can find any tertiary butyl acetate in the environment that you're looking in. I don't think it occurs, I don't think it appears on an 8260 or a standard GC analysis. So you might have to have someone develop an appropriate analysis for it. But if tertiary butyl acetate is present, um, and you know what the ambient pH is, you can use that information quite readily to start to answer that question. There are isotopic methods that can be used to discriminate the sources of TBA in an ongoing biodegradation process. And that's something that we've worked on a bit, and we hope to have a paper out published on that later on this year. And what that would enable you to do is discriminate, or it's designed to discriminate between TBA production that occurs from MTBE and isobutane. And it would work just as effectively for tertiary butyl acetate as well. We haven't included that in our study, but it would be possible to do that. And if that's a concern and somebody wants to, my email address, actually my email address is being modified, but that one will still work. If the person who's asking that question would like some more detail about it, I'd be happy to share it with them.
Okay, great.
But yes,
Can
there are
TBA,
tools.
I'm sorry. Can TBA be toxic for bacteria? They have a site with 2,000 ppm of TBA and low degradation rate, so do you think that could be a toxic effect?
I would be surprised. 2,000 ppm is not that much, really. You know, when we start dealing with toxicity issues for alcohols, we are typically measuring things. You know, so if you want to destroy bacteria in the lab, you use a 70% volume mixture of ethanol because that dissolves membranes and it messes up bacterial proteins. If you're way below that in the ppm level, I would be very surprised if you could account for it by a toxicity issue. What would be much more likely, in my opinion, would be some restriction, environmental restriction. And one thing that I didn't talk about in this presentation, but in any detail, but is perhaps relevant, that very last reaction where you are looking at the conversion of 2-hydroxybutyrate to 2-hydroxyisobutyrate to 3-hydroxybutyrate, that is catalyzed by a mutase enzyme. And that mutase enzyme has a absolute strict requirement for cobalt. I don't know what environment that you are, the question is coming from, but if it was an exergy reactor, for example, one of the things I would look at would be, is there sufficient cobalt ions present for this physiological activity? It might be harder to do in the field, but that is, you know, very clear constraint on the ability of organisms to biodegrade TPA. If there isn't sufficient cobalt, that process will just slow and stop. And we're not talking about pounds of cobalt here. We're talking about trace nutrient levels of cobalt.
Okay, so if we have both TBA and MTBE that are present under oxic conditions, can you tell which one would be oxidized first in the most likely event? Would it be TBA?
Well, that's going to be dictated by which organisms are present. Um, I, the I would hate to predict which way it would go. I can tell you what happens in certainly in the organisms that we study. TBA goes second. Um, but that is, we typically have not run studies or looked at studies where we start with an equimolar, for example, concentration of those two reactants. So it might be a bit of a dangerous extrapolation. Um, the thing you kind of need to bear in mind is that irrespective um, of the mechanism of biodegradation, under aerobic conditions, MTBE degrading organisms are growing on TBA. TBA is actually what they get all their energy from. The conversion of MTBE to TBA is an energy requiring reaction. So you might, although I can't off the top of my head point to any evidence, concrete studies that have described it, you might predict that TBA would certainly start out faster than MTBE. Um, there are, until you know what type of microbial process was responsible for getting rid of the TBA, I think it would be difficult to predict that. A co-metabolic system would give you a very, very different performance than a system based on metabolism, for example. Um, if you are in an environment where you still have a source area, you still have free product, or you're in a source area where the concentration of organics from gasoline is very high, I would predict that you would have initially a co-metabolic process that would convert your MTBE to TBA, in which case the MTBE would go first and then the TBA would likely go second. If you don't have other gasoline co-contaminants present, that would change the type of microbial process that you would expect to be degrading those compounds, and the answer might be different. So perhaps the person who asked the question would like to give me a little bit more detail. I'd be happy to send an email back and give you my best thoughts on that and any examples in the literature which might back up what I'm suggesting.
Okay, one last question. Can you comment on the successive degradation of MTBE and TBA where BTEX and total petroleum hydrocarbons have been first substantially degraded before MTBE is being degraded at any appreciable rate?
Okay, so again, that's a somewhat related question. Certainly under aerobic conditions, one of the most consistently effective treatments is adding oxygen. I don't think people have really asked how is the compound biodegrading, how are MTB and TBA biodegrading. That might be crossing the T's and dotting the I's and far too academic uh, for a field application. But if there are gasoline components present, you're doing it aerobically, you are likely to have a co-metabolic process. If you don't have gasoline components present, you, I think, are going to be much more reliant on MTBE metabolism. In other words, organisms like PM1, L108, being able to grow on those compounds as independent carbon sources. When you go to the anaerobic environment, um, I think it would be accurate to say that conventional wisdom says that the anaerobic degradation of other gasoline components creates the conditions that allow anaerobic MTBE degradation to TBA by acetogenic organisms. An MTBE only spill, unless there were very, very high concentrations of MTB present, just don't, the compound is not sufficiently intrinsically biodegradable to convert the environmental conditions that are necessary to allow acetogens to thrive. So gasoline, in many aspects, is an important driver of MTBE degradation. It's providing the carbon, it's providing the fuel, it's providing the energy that is going to alter the environmental conditions to allow the degradation of MTBE and TBA to occur. So yeah, I think you can't look at MTBE and TBA without considering how much gasoline, how much gasoline biodegradation products are still present, because that's what's manipulating the environment to allow those compounds to be degraded themselves.
All right, that was great. Well, thank you very much, Mike. There are a couple of other questions that we didn't get to due to the time today, but what I'll do is email those to you and then email them out to anyone that's interested in the answers.
Sure.
But thank you again, Mike, for doing this presentation. It was great as always, and we really appreciate that you would do this for us. And we hope that everyone on will join us again in July for our presentation, and look forward to talking to you more then. All right, thank you so much, Mike.
And thanks for everyone for sticking around and listening. Appreciate it.
All right, thank you.
Bye.